All right. Any questions so far on this, folks? Okay. We're going to top things off today by mentioning a few basic categories of transcription factors. Now, there's a whole bunch. It turns out, although there's over 1,000 different genes for different kinds of transcription factors in our genome, I would say 20,000 or so, well over 1,000 different genes code for certain types of transcription factors. Some very specific, some much more broad-based. But fortunately for us, those enormous numbers of transcription factors we can place in a couple of different categories based on certain structural features they have or on how they bind to DNA. So we can put them in about 5 different families. Now it's likely that if you go way, way back into the earliest part of life, each one of those families may have originally derived from a single ancestral protein. So maybe you had in some of the earliest cells that are too old to be in the fossil record, maybe you had just a handful of different transcription factors that regulated the several hundred to a couple thousand genes that the earliest cells may have had. There's a debate. What's the minimum genome? There's a big debate on that. What's the minimum size genome to make a living cell? It looks like it's about 500 genes. Okay, so a limited number of transcription factors. As organisms evolve, got more and more complex, you have duplication, divergence of these different genes, and then you get families of related transcription factors that can take specialized functions. So now we got like 1,000, but they fall into basically kind of about 5 different categories. Most of them are put in these families by certain key structural features. One of them is what we call the Helix-Loop-Helix proteins. And they have a certain structure, an alpha helix, a kind of coiled loop of protein, like so. They have that particular type of structure, and this is what actually binds the DNA. Change the sequence of these amino acids here, and that determines what sequences the DNA will bind. In fact, people have done experiments where they've done things with genetic engineering. Well, take a certain transcription factor and alter the sequence, and you end up binding to a different section of DNA, a different enhancer. So you can do that kind of thing. Okay, so that's our DNA binding region. For the most part, these guys assemble together. They bind together, form a dimer, just means 2 proteins together. Okay? And that's the actual active version of it. Now, they don't have to bind to 2 identical copies. One of these proteins could bind to a different helix-loop-helix protein. And that's going to affect what kinds of genes are being activated, things like that. And then usually, that dimer will bind to yet a third and sometimes even more one of these members of the family. And that determines what they do to genes, what enhancers they bind to, and things like that. One example of that, the myo-D proteins that are critically important in turning cells into muscle, and our infamous widely distributed oncogene, or at least when something goes wrong, called MYC. That's distributed in many different cell types, and when it's misregulated, it can stimulate abnormal cell division, which means cancer. That's a good thing to have. Okay. All right, so that's another kind. what are known as the Leucine Zipper proteins. Now, they're called that because they have these stretches, these kind of long stretches of amino acids that's loaded up with the amino acid leucine, which I'm representing as L's. And then what happens is a second member of it binds together. So it kind of sticks together like a zipper. And that's a leucine zipper. Yes?
How do you spell that? We can't figure that out.
Oh, this? Oh, Leucine. Sorry. Now, leucine is one of the 20 amino acids. And those zipper regions are rich in the leucines stick together, holding the two proteins in a dimer, and then it basically straddles the DNA, sort of like this. Kind of like, kind of like riding cowboy here. They sit on the DNA. Yes. Hi, ho, transcribe. Okay. All right. So these are leucine zipper proteins. This is another family. The key things, they stick together with these extended things of extensions that are rich in this amino acid leucine. And leucine's hydrophobic, by the way, so that's why they stick together. If you're in an aqueous environment and you have a whole bunch of hydrophobic amino acids sticking out the side, you want to get out of that hydrophobic environment. Good way of doing it is stick with your partner, right? The same thing. So that holds these guys together. Okay, so that's another one. A very common family, the zinc fingers. They're called that because the DNA binding region of a zinc finger protein is several loops of amino acids that kind of stretch out like a series of fingers. And in there, each one of them is a zinc ion. And the zinc ion holds those loops together, stabilizes those loops. And then the different zinc fingers kind of caress the DNA. Oh, will you please transcribe for me? But the zinc finger is actually a DNA binding region. And there are quite a number of these types of things. Steroid hormone receptors are a good example. They're widely used, many of these zinc fingers. Okay. And two more.
Was there an example of a Leucine Zipper?
Nothing that would strike anybody's fancy. There's a number of these things. There are some examples, but they're just not, they're not garden variety ones. I figured some of the ones I mentioned and stuff. Okay, so that one I kind of left out because something nobody's heard of, or at least you guys haven't heard. Okay, now another one. What are called the homodomain proteins, sometimes called the homeobox proteins. These all have a certain sequence of about 50 amino acids called a homeobox. And although the sequence has a lot of similarities from one protein to the next, the differences determine exactly just what enhancers they bind to. Okay, now some of the homodomain proteins, the ones with the homeobox, are some of the most important developmental proteins around because they were first discovered in weird mutations in fruit flies, where you'd have things like absence of eyes, absence of wings, legs growing where their antennae is supposed to be, missing segments, duplicated segments. They traced it down in the 80s to exactly what genes they were. They found this class of DNA binding proteins. They all had this certain little region they called a homeobox. And there you had it, Kevin. Of course, we have plenty of those in our own genome, mostly variants of the ones you find in fruit flies. But best example of these homeobox proteins, homodomain proteins, are the Hox genes, or at least the proteins that these are coded for. These Hox genes in all animals organizes the anterior to posterior axis. In other words, these are the genes that tell you what is tail and what is head. Simple organisms have only a few to a handful of Hox genes. For instance, sponges. Look, let's face it, sponges don't have much of a head to tail axis, right? They don't need too many, so they only have 2 Hox genes. Us and other mammals, we've got 39 of them. Each one specifying the formation of certain structures, especially sort of like the main axial or body skeleton, the spine, no column, the ribs, stuff like that. This neurotube, in other words, the brain and spinal cord, those Hox genes play important roles in that in our development. And that's the best known example of these. These are the genes that literally allow you to tell head and tail apart. And this is something that's universal in all animals. It's just the more complex ones have more of these genes. Okay, last one is a close relative. It has an extra DNA binding region. They're called a POW. I think that's how it's pronounced. They're actually a subset of the homeobox proteins. With an additional DNA binding sequence.
With an additional, what sequence?
Additional, called POW, P-O-U. It's another DNA binding region. So it has 2 different DNA binding regions, the homeobox and this so-called POW sequence. Okay, oops. Okay, the example of that, we might run into this guy a little bit later, is a protein called OCT1. OCT1 is a transcription factor that regulates transcription in a huge number of different genes. It's one of those really broad-based ones. If you look at probably a good portion, maybe half or a quarter of our genes, you'll find OCT1 binding to them. They may have different effect depending on what else is floating around, but it's one of these really broad-based transcription factors that affect transcription in numerous different genes. So fortunately for us, all the transcription factors we've identified do fall into these broad categories. Mostly, you're looking at mostly structural similarities. How they assemble together, what kind of things, the DNA binding region and stuff like that. So it's quite possible these 5 families, it sounds like New York City, right? 5 families, okay. Possible these 5 families all came from a couple, 5 different ancestral godfather proteins. We have an offer you can't refuse. Way back in that they're actually descendants of single ancestral transcription factors way back at the dawn of life. Okay, so that's what we see here, and this is a good time to say, hang it up, and I'll see you guys in the lab fairly shortly. Once again, please forgive me for trying to put this thing together.